bacteria have an enormous potential to adapt and evolve in very small time scales because of the small generation times, and that can be seen from this timeline where resistance to most antibiotics has been seen to develop almost immediately after they were introduced. We are interested in the antibiotic kanamycin. It targets the ribosome, and it blocks translocation at various states. Um, so a direct mutation in the target of the antibiotic, that is the 16S rRNA gene, is possible, but not accessible to E. coli because there are multiple copies of the gene. So we were curious to try and understand this using E. coli as a model organism, whether growing E. coli in sublethal concentrations can lead to gain in tolerance, and if so, what are the mutations conferring tolerance, and what are the trajectories taken by such evolving populations, as in what sort of mutations, how do the abundances of mutations bearing various levels of tolerance change with time, and also are these trajectories dependent on the concentration used. So just very quickly, we use the MIC as a readout for tolerance, and we have taken around two concentrations. One is 25% and the other about 50% of the lethal dose. And this is our regime. We use batch culture to grow E. coli either in the presence or absence of antibiotic. We transfer populations every 24 hours. And maybe what I'll do is show what are the sort of differences we see at two different concentrations at particular time points in this evolution experiment. So in the 4-can regime, we see that, okay, so what is plotted here on the x-axis is growth in the absence of antibiotic, and on the y-axis is growth in some concentration of antibiotic. So for the 4-can regime, we get really homogeneous populations, but for the 8-can populations, we also start getting individuals that are tolerant, but that have also incurred a large cost. They cannot grow as well in the absence of antibiotic. So we see like different subpopulations, at least on the phenotypic level. Genomic level, it seems there are even more further subpopulations. Okay, so and this disappears subsequently, and we have done population sequencing or sequencing of individuals derived from these populations to find out what the mutations are and what are the dynamics in this population. That's it. Thank you.
Mutators are individuals with higher mutation rate than the wild type, and mutators have lower fitness because most of the mutations are deleterious in nature. In Lenski's long-term evolution experiment, it's observed that mutators will get adapted, mutators will get fixed in populations which are adapting. So in this diagram, it's taken from Lenski's paper. The white colored bars represent wild type, black colored bars represent mutators. Very recently, it's observed that mutators can revert back to wild type. I am considering a population which is sitting at the fitness peak, adapted population. So I can work in the regime, this particular regime, so that I can neglect beneficial mutations. If the population is here, there will be beneficial mutations, and I'll have to include hitchhiking, the process by which mutators will get fixed in the population. This is the reason why mutators take over the population in Lenski's long-term evolution experiment. In my model, I consider asexual haploid population, and the population size is constant. It's represented by n. It's a finite population. Fitness landscape is multiplicative. Mutation rate of mutator is v, that of wild type is u, and v is always greater than u. The conversion from wild type to mutator is always greater than that of reverse conversion. And my work is the extension of work done by Kavita Jain and Apoorv Nagar. In that work, the only wild type to mutator conversion was included, and they found out that even in the absence of hitchhiking, mutators will get fixed in a finite population. So I want to know what is the effect of including the reverse conversion from mutators to wild type. In a finite population, in finite time, there is transitions from mutator dominated state to transition between mutator dominated and wild type dominated state. Tn represents the time wild type dominates, Tm represents the time mutators dominate. You can see Tn increases with n is population size, Tm decreases with population size. So when I average over this dominance times and plot it as a function of population size, what I see is Tm, the red colored curve, decreases and Tn increases as a function of population size. There exists a value at which these two curves intersect, which is a critical population size, above which wild type dominate and below which mutators dominate. And so below which there will be lower fitness, above which mutate wild type dominate and population will have higher fitness. I have obtained a formula for this particular value of NC in strong selection weak mutation regime in terms of parameter. For reasonable values of these parameters, in the case of E. coli population, critical value of population size is found to be of the order of 1000. And this is a conclusion future work. Analytic expression is well matching with the simulation result. Currently work is being done in the weak selection strong mutation regime. Beneficial mutations and epistasis can be taken into account in my work. Thank you.
Hi, I'm Aparna, and I'm from Center for Ecological Sciences, which is just opposite the physics department. And I have a very short presentation because I have not started my work yet. So I'm just going to present my hypothesis. I'm broadly interested in systematics and historical biogeography, and in particular lizards. The system that I'm studying is Hemidactylus geckos, and geckos mainly because it's really easy to get permits in India because nobody cares about them much. This is a particular gecko called Hemidactylus brucei, and it was thought to be a single species, but some exploratory work that I did, so everything except the blue box is what was known as Hemidactylus brucei, and within the blue box are actually morphologically divergent different species. So each tip within the blue box is a different species. So you can look at how much variation there is within what was thought to be brucei. And what is more interesting is each of these have very different habitat specificities. So two of these clades, which are putative species, are human commensals. One is a rock-dwelling gecko, and one is a ground-dwelling gecko. So this is even more interesting in the context of the landscape that you find them in. I'll come back to this question, but they are found broadly in the dry zone of peninsular India, which is just the triangular part south of Narmada. And this is, if you just zoom in, all the tiny circular things that you see are rocky patches. So these rocky outcrops are naturally fragmented. So the habitat of rock-dwelling gecko is naturally fragmented as opposed to ground-dwelling gecko, which is more or less continuously distributed. And the human commensal gecko is another interesting group because they're thought to be moved around by humans. So my broad objective is to understand how this habit of being commensal or not, or the habitat, whether it's fragmented or continuous, affects the phylogeographic pattern. And my hypothesis is something like this, that if you look at isolation by distance, because the rock-dwelling geckos have a fragmented habitat, they'll have a greater isolation by distance as compared to the continuously distributed species. And human commensal geckos, if they're moved around by humans, might not show much pattern. Thank you.
Hello, am I audible? Hello, I'm Aparna. I am in National Center for Biological Sciences. I work in Dr. Deepa Gash's lab. So today what I'm going to talk to you about is some work that I have been doing at looking at patterns of nucleotide change under selection. So the thing is that as we have been learning in this course as well, that when you look at very simple models of nucleotide evolution, they assume that there are no well, the nucleotide change rates are constant, but we all know that that's not true, and there are different biases in the inherent mutational rates themselves. For example, one of the mutational biases that was also talked about was that transitions are much more common than transversions. And so now if you look at different bacterial classes, you can see that bacterial classes have their GC content fairly conserved across within the class itself, whereas it varies quite a lot between classes. And recent studies have shown that if you like in bacteria, the basal mutational rate is biased from GC towards AT. That is that most mutations, like a higher number of mutations would happen from GC to AT than the other way around. And if you believe that is true, then there are two questions that arise. How do you even begin to have a high GC rich genome? And secondly, how do you maintain a high GC genome? Even if you have it by some drift or something, over time you should lose the GC from your genome. But that is obviously not true. And there are many reasons why, like reasons you can provide to counteract this argument. And two basic reasons you might think of is maybe there is some selection on the GC content itself, or maybe that so basically what I have done is I have looked at different experimental evolution studies, and I have looked at all the changes that have happened under conditions of selection that are known to us. So we know what the selective pressure is, and we know what the selective advantage of that particular mutation is. And then I have tried to look at, okay, so what are the more, are there any biases that we observe under selection? Because all the changes, all the biases that I have talked to you about till now have been reported under conditions of, they are measured under what is termed as neutral conditions, so that there is no selection. So are these changes different when you put bacteria under selection? And also whether these mutations themselves have a higher fitness effect when you compare across the two classes. So till now what I have found is grossly there seem to be no biases that are seen in all the studies that I have looked at, but when you look at individual studies separately, you see some biases.
So hello everyone, I'm Ashutosh from Jawaharlal Nehru University, and I'm working with Dr. Supratim Sengupta. So he has now recently moved to IISc Kolkata, and I'm working on the origin and evolution of the genetic code. So in this problem, this particular problem, we looked at the physicochemical hypothesis, and we tested if it is able to explain the emergence of the standard genetic code. So as we all know, genetic code is a set of rules by which information in the genome is translated into proteins, and I have shown here the standard genetic code. It's a mapping between 64 codons and 20 amino acids, and there are 3 stop signals, and various hypotheses have been proposed to explain the origin of the standard genetic code. One is the frozen accident theory given by Crick, which says that the standard code it was just an accident, and it requires no explanation. The physicochemical theory says that the standard code is a result of selection to reduce the harmful effects of mutational and translational errors. And the correlation theory says that the code co-evolved with the pathways for amino acid synthesis, and the the 4-column theory, it says that in the early stages of code evolution, there were only 4 amino acids encoded, and gradually new amino acids were incorporated in the genetic code. So, to test the physicochemical theory, we are using a finite population model of code sequence coevolution, and we are using the code proposed by Higgs as our starting code, and with that code we are generating a set of plausible primordial genetic codes by reassigning codon blocks to new amino acids. And initially each code is translating a fixed number of identical DNA sequences and mutations. We are making mutations as per the Jukes-Cantor model, and we are using this formula for the calculation of fitness of each sequence in our population. And the population for the next generation is obtained by selecting sequences in the population in the current generation with probability proportional to their fitness values. And once the sequences have adapted to their respective codes, sequence sets are made to compete with each other till one of them gets fixed in the population. And we are calculating the fixation probability of the codes which are getting fixed using this formula. And what we find that in a comparison with randomly generated codes, the code which is most consistent with the standard genetic code, which I have shown here, CSGC. This is a 10 amino acid code which is most consistent with the standard genetic code. So it was found to have a very high fixation probability. But when this code competes with a constrained set of codes, which consists of physicochemically optimized codes, its fixation probability was of the order of 10⁻². And so I will just put the conclusions. So our results indicate that the selection of the SGC over alternative codes which differ marginally in the degree of optimality cannot be explained solely by the physicochemical hypothesis of code origin, and standard code could be a result of combination of adaptation and frozen accident. The other references, thank you.
Hi everyone, I'm Avni, and I know the topic sounds very lengthy, but broadly speaking, I work on sexual conflict, especially in flies that are fly populations that have been selected in the lab conditions for specific traits. And I'll just introduce these terms. So sexual conflict is basically conflict that arises between the sexes, and that essentially happens because males and females differ in the ways in which they maximize their reproductive fitness. And therefore, one can imagine that traits that might be beneficial for one of the sexes, because the two sexes interact, can simultaneously also harm the other sex. And that would result in conflict between the sexes. And if that happens, the other sex is expected to evolve some sort of defenses against these traits. So in case of fruit flies, what we know is that one of the ways in which this happens is that the males tend to manipulate the female's physiology in a number of ways to maximize their fitness, and the females therefore evolve defenses against that. And size, body size, plays a big role in this. The second thing I want to introduce is the populations that I work on itself. And we do lab selection experiments, which basically means that we select populations for very specific traits. And the expectation is that although we select for specific traits, there will be correlated changes that will happen in the population. And responses will be seen in traits that are not directly selected for. So in the populations that I have worked on, one of the things that evolved was, so I selected for faster development in flies. And one of the things that evolved was body size in those selected populations, which basically reduced. So the lower panel shows the body size of males and female fruit flies in the selected populations. And you can compare that with the top panel, which shows the body size of the ancestral controls. of these populations. And I wanted to see, so there were earlier indications that some earlier work had suggested that there might be changes in the levels of sexual conflict in these selected populations. And therefore, I wanted to see what the contribution of body size reduction was to this. And for that, I used something called JC there, which is basically a body size control, which is basically a population of flies, or not a population, flies that are genetically belong to the same background as the controls, but are of the same size as the selected populations. And the readout that I used to do this was the fitness loss that the females experienced in terms of increased mortality post-mating. And I can just draw your attention to this particular bar, which basically shows that the selected females did not die any more than the control females when they were mating with the body size control males, which basically means that when you reduce the size of the males are not as manipulative, and that is indicative of a lower level of conflict. And this is one of the more interesting results from this one experiment that I had done. And additionally also, if you reduce the size of the female, which is seen here, if when they mate with a male that is very vigorous and is very manipulative, they don't die anymore. So this basically this one experiment showed that it matters in case of males to be big if they have to manipulate the females, but the females are, there is this, again, such traits are very, are more dependent on genetic background. Sorry.
Hello. Yeah, working on human cooperation at Center for Behavioral and Cognitive Sciences. I'm going to present what I do. Yeah, the problem is actually in cooperation. This is a problem. Under what conditions will cooperation emerge in a world of egoists without central authority? It is a philosophically also interesting question. Without central authority, is it possible to get a cooperation? This answer is partly addressed by Axelrod. Why we need to study cooperation? Because understanding cooperation, we can also design the societies that will give you maximum cooperation. Okay. Let me explain what is cooperation. Cooperation means, suppose if helping somebody, I have to incur some cost. C, the guy who is receiving help, he gets a benefit B. That's a mathematically right as a cooperation activity. If the two individuals, for example, X and Y, interacting each other, if X cooperates Y, he loses minus C, he gets plus B. Suppose X and Y, X is defecting Y, he doesn't get any payoff. Suppose X and Y, X is cooperating with Y, he loses minus C. Suppose Y also cooperates X, you get B. In this case, you get the B minus C payoff. I can define this interaction by writing the game. It's a 3D game. Player 1, player 2. First player cooperates, second also cooperates, you get B minus C. If the first player defects, second player cooperates, you get the minus C. If this guy cooperates, you get defect, this is 0. This is a generally standard game people use to capture the logic of cooperation. If I analyze by looking at player, what they do according to given player strategies, they end up with a defection. The second player also uses the same logic, he also end up with a defection. I'm not analyzing this, just I'm giving. The DD, defection, is the generally stable strategy. There are a lot of mechanisms available in the literature. One is the kin selection. Kin selection is explained in terms of biological relatedness with other individual. The other two mechanisms are direct reciprocity and indirect reciprocity. Direct reciprocity, you scratch my back, I will scratch my back. Depends on that. Generally, this direct reciprocity works very well if the interactions happen between the same people again and again. Okay, what it was, I interested in indirect reciprocity, where actually I help somebody, they get the help from some other people. Here, the information about the cooperation driven by the repetition. Suppose So A help B receive help from somebody else. There is reputation information passed to the gossip. I try to model how this gossip transmits information by using this mathematical formula. What it was, I don't have much time. What it was, I able to figure out what is when I know about the information about particular individual reputation, how I know that the information is credible. That I capture by this formula. The P gives me that probability of cooperation if I know other individual reputation. I'm not explaining this. Let's finish this one. Yeah, here I shown some simulations. Actually, these are the right one is the simplex. Yeah, I shown with different information scenarios where this trajectory will goes. Yeah, there is different results. That's it. That's fine.
Hello, I am currently working at IISc with Dr. Prasad. So I work on the evolution of the male reproductive behavior in response to selection for cold shock resistance. So there are so many environmental states which affects the physiology and behavior of the organisms. So why it's a cold stress is matter for the organisms? Because especially for the insects, insects are the phycallothermic organisms. They cannot maintain their own body temperature like the warm-blooded animal do. And so for that I use to study this and this temperature especially decides the distribution of the organisms and how you do these things. And the previous study they reported that look at the lifespan evolutions, but nobody looked at the reproductive parts of the in response to the cold stress. So we use the laboratory approach. Here is our baseline populations. We create from these baselines to the selected lines and here control lines. So in the baselines, in the selection lines on 12th day post egg collections, we transfer the vials flies into the empty vial and expose them to the minus 5 degree temperature. This is the only single event difference between this control and the selections. And then we look at after several generations of the selections on the male reproductive behaviors. So this is the experimental designs. The FSB and MCB, we collect the virgin male and females, for the virgin males on the 9-10 days, and the 12th day we expose them to minus 5 degrees and then we allow them to recover at different time points like the 4 hours, 12 hours and the 30 hours. Then we look at the behaviors, mating latency, copulation durations, male fertility assay and the mating number. So these are the results. So mating latency, the selected lines males is the black one is the control and this empty one is the selected lines. The selected lines mating latency is the selection aspect is significantly reduced in this case of the mating latency, but there is no difference in the copulation duration between the control and the selections. The number of mating is increases in the selected lines compared to the control. Yeah. Okay. And proportion of the unhatchability. So how we did this after copulations, we transfer the females to lay the eggs in 48 hours. We check whether they eggs at least the one female produce the fertile eggs. So, and so my conclusion is that so evolution of these male reproductive behaviors is the correlated response to the cold stress resistance. Thank you.
Hello. So I'm doing PhD. I've joined last August in JNC as a molecular mycology lab under Kaustubh Sannal's supervision. So the title of my talk is Evolution of Centromere in Pathogenic Yeast Species. We deal with pathogenic yeast species
including Candida albicans, Candida tropicalis, and another species Cryptococcus. So I guess there are a lot of non-biologists, so I will explain some terms. So kinetochores are proteins which associate on centromeric DNA, and centromeric DNA is the part where these proteins assemble. So why centromeres are required? Because when the genetic material is segregating, they are needed to be held at one part and to be fetched apart to two folds. Okay, so these are the part where kinetochore proteins associate and they perform this function. So if you look at a chromosome, here is the centromere and here your kinetochore proteins associate, and it looks like this when the microtubule binds to your kinetochore proteins. And this is the organism I work on, Candida albicans and Candida tropicalis. So to give you a hint about the complexity of these kinetochore proteins, I have given one image. So many proteins are there. Okay, so yeah. So I'm looking at a problem where I will tell you some facts like so if you look at the relationship between these four species, this is a basic budding yeast Saccharomyces cerevisiae, which is also known as Baker's yeast. And these are two organisms we are working on currently. And Saccharomyces pombe is another organism in which structure of centromere in terms of DNA sequences is like that. If you look at cerevisiae has a genetically controlled centromere, and in pombe has a repeat associated centromere which has repeats which are heterochromatic in nature, and this heterochromatic information is essential for its centromere function. But when you look at Candida tropicalis, we find a very unique situation where it has a similar centromere like pombe. Also, it is closely related to Candida albicans. And so another fact I will tell you that this centromeric heterochromatic information is dependent on the RNAi machinery. So if you look at the key RNAi proteins in these three species, you will find that Candida tropicalis doesn't have the dicer as well as Candida albicans. So, but these three proteins are required for their function. And so I am looking at three possibilities, which are albicans and tropicalis centromeres are not heterochromatic. So there are also other possibilities, but I am currently looking at this first objective. So I am trying to see if it is really heterochromatic or not. Okay. So I am looking at a problem which is related to all these components. I am trying to figure out the relationship between all these components like RNAi, small RNAs, DNA sequences, and heterochromatic in relation to centromere function and their implication in pathogenic budding yeast. So I am what I am doing is okay. So right now I have integrated one transgene in the centromere and looking at their expression in different selection mediums, and I have tagged this protein NPA, which is known to be deposited in centromeres, and give the centromeric identity. So if you look at this, their localization, they are specifically going to the centromere and giving the centromere identity. Yeah.
I'm audible? Yeah. Okay. I'm Manashwini. I'm going to tell you about one of my experiments, which I did it using Drosophila melanogaster populations, which were selected for extreme levels of crowding during the larval stage. So, which is like one of the really high selection pressures when, I mean, in a Drosophila life cycle, you have this egg stage to larva, to pupa, to adult. So, when there is a high intensity of population around you, so the first thing that comes into mind is that you are selecting for greater competitive ability. So in a high dense environment, competitive ability is a trait that we look for. So and moreover, competitive ability is not just one trait, it's the combination or an effect of multiple traits that gives rise to, to see a response. At least in Drosophila context, foraging behavior, tolerance to waste, your efficiency of converting food to biomass. So there are just multiple traits that one can think of that the population is evolving in. Now, in high density environment, it will be it will be better if you can acquire resources faster, hence increasing your feeding rate. Or maybe you can increase your level of tolerance, because over time your culture might be very inhospitable, so it will be better if you have high tolerance level. Or maybe you can adapt some other strategies where you become more efficient, maybe by developing faster or by reducing body size, which improves the growth rate. So essentially the idea here is the relative importance of this fitness related traits depends on the ecology. So which of the traits will lead to a certain trajectory depends on what ecology you are in. So here I'll tell you about the total food available or the larval density, or both of the interactions between these two factors is leading to a kind of trajectory. So before going to the objective, the background of starting this experiment was of two different sets of Drosophila populations which were subjected to different levels of crowding. And essentially, as you can see, so this MCU population, which is the population that I work on, means the melanogaster, crowded in the larval stage, uncrowded in adults. So as you can see, 600 eggs in 1.5 ml food versus the other population, 1200 eggs in 6 ml of food. So clearly, clearly you can see that there is a very high difference in the kind of amount of food versus the eggs. So here I wanted to test this hypothesis by creating some environment in which you put the eggs in all kinds of regimes. And so what I found was, obviously when you go from low to high density, your survivorship decreases. Moreover, you see that it is not a linear thing. I mean, if you go on increasing the density, the survivorship in the 1203 ml, which is a mediocre density, it is least in that, whereas in the 6 ml, it is higher. Moreover, it is very interesting to see the pattern of how they close over time. When they have more food, it seems that they take a longer time, and there's a wider spread in which how they close. Similarly, that is seen in the body size. So the selected populations which are selected for crowding are essentially smaller. And again, there is like the early flies seems to be bigger than the ones which are coming late, and so on. So it has a very interesting pattern by itself. So yeah. So it is definitely an interaction of both the food level and the larval density, and not solely one factor is contributing. That's all. Thank you.
So I'm Meghna. I work in Dr. Uma Ramakrishnan's lab at NCBS, and we're working on a project to do with connectivity between tiger populations in the Indian subcontinent. So just a brief background, which we've gone over pretty much, but when populations diverge from an ancestral population, when they split up, they undergo divergence over time, especially due to drift. But how much divergence is going to happen is reduced by the amount of gene flow that might be happening between them. But overall, when you have small populations which are especially isolated, it is not a great thing for the population, because they accumulate effects of inbreeding and genetic drift, eventually losing variation, and therefore they have lower fitness and viability, and this feeds into becoming an even smaller population and may even eventually lead to extinction of the population. So but why this is interesting in the context of tigers is because in the Indian subcontinent, tiger populations are split into many small populations. So it's like a meta population where you have sources and sinks, and some connected, some isolated populations. So you can really look at gene flow and variation within these populations. So this is a slide which was many things, but all I want to say is that there are of the extant subspecies of tigers, only one is present in the Indian subcontinent, which is the Royal Bengal tiger, and that is the most numerous species, with also it has been shown the most genetic variation. But also, as you can see from the color difference, the historical distribution showed that tigers were pretty much spread out evenly throughout the subcontinent. But in recent times, maybe over the last few hundred years, tigers have really shrunk in their ranges and occupy now very fragmented, patchy landscapes here. So in a study that was done in the lab previously, they basically collected samples from historical tigers present in historical times, so from museum samples especially, and they wanted to see if there's any change in connectivity between past and present. So looking at the museum samples, basically this is the result of a structure plot put in graphically, but to look at if there's any substructuring. In historical samples, they picked out just 2 genetic clusters from the entire distribution. Whereas if you move on to the present and remove all the historical samples, the northern cluster splits into 2. But more interestingly, the southern, all the populations in peninsular India still appear as one cluster, which is interesting. I mean, at least for the northern populations. But if you zoom into the southern region, you can see that the populations actually, and these green patches actually reflect protected area network in the Indian subcontinent. They're really spread out, they're small, and they look isolated, although there are some forest patches. So it really, you might wonder how does this appear as one genetic cluster when it really looks like, it might actually be quite fragmented. So this is not really work that we've started yet, but we're just sampling. But what we want to look at is, does that central population in between act as some sort of a conduit for movement between the upper cluster and the lower southern landscape? And what is the pattern of substructuring that is present in this landscape, and also what landscape features themselves, natural and also human presence, how does that affect, or how is that mapped on to this region?
Am I audible? Hi, I'm Rudula from Deepa Agashi's lab at NCBS. And today we all heard about how synonymous mutations are selectively nearly neutral. But some recent studies have shown that that's not the case. And in an effort to understand how that comes about, Deepa in her postdoc created lots of alleles of an essential gene which had lots of synonymous mutations. And what she found was that in spite of the fact that these mutations were synonymous, these alleles did very poorly under selective conditions as compared to the wild type. And what she also showed is that this low fitness was due to reduced gene expression and protein production from these alleles. And she also showed that when you increase the expression from these alleles by putting them on under an inducible promoter, growth defect was rescued to a large extent in most of the cases. What she did later was she evolved these strains which bore these alleles in the lab, and she found that single point mutations had arisen in each of these cases, in each of the alleles. And so this table shows you basically that these point mutations arose in the beginning part of the protein coding sequence, and in some cases in the non-coding regions. And these what's cool is that these mutations are repeatable across populations as well as across independent experiments. So what I'm doing in the lab is to try and figure out exactly how these mutations are able to confer fitness back to these strains. What I've done is I have taken the mutation from the evolved strain and stuck it back into the ancestral background and try to figure out what happens. So what I see is that when you put the mutation back on the ancestral background, the fitness of the mutation is almost as good as the evolved strain, whereas when I do the reverse process, that is the ancestral gene in the evolved genomic background, it has almost zero fitness. What I've also done in the lab is to check for protein levels from these so-called allele swap strains, and I've shown that the evolved mutation also leads to an increase in protein production as compared to the ancestor, whereas the evolved background really has no effect on protein, which correlates with the fitness. That's it.
Hello. Anyways. Hello. Yeah, good afternoon. Myself Mukesh Thakur, and I'm working with WI as young scientist. I'm going to talk something exciting on bears. Bears are largely distributed in the northwest Himalayas and the hills of northeastern India, and I worked in the area which falls somewhere in the Kashmir Valley. It's called Datigam National Park, which falls in the state of Jammu and Kashmir. This was my study area, the Datigam National Park. This is somewhere around 100 square kilometers, which we divided by putting a grid of 2 by 2 kilometers, and we put up some hair trap stations. This was the design of a hair trap station. So just to collect the samples, that non-invasive samples, we put this. We made such kind of hair trap station and put into the this our protected area. So this was kind of design. 3 bar fencing were tied up with the 6 posts to make a hexagonal kind of structure, and we put it into the different locations. And we used some rotten meat or some fruits to lure the bear towards the hair trap station. And all the hair trap stations were monitored using a camera trap station, which was set up just right in front of it. And then all those 23 hair trap stations were monitored twice a day to carefully collect those all trapped hair samples. So these are some of the pictures which we got during our study. And apart from this, we also collected some reference blood samples. Like we radio-collared 7 individual bears for studying their movement pattern. And in spite of that 7 individual bears, we collected 11 blood reference samples from those rescued bears, which were rescued by the forest department from the conflict sites. So we selected this 20 microsatellite markers from the previous study conducted on different bear species. And this is the diversity estimate for the blood reference samples only, with 18 reference samples. And based on several parameters, the person who is working on population genetics, particularly on microsatellite, they must be knowing that what are the parameters we use for individual identifications. So based on their high success rate and low amplicon size, and the probability of identity and low genotyping error like allele dropout and false allele, we selected the panel of 7 microsatellites, which is highlighted in the above of the table. And then we have identified 109 individuals. Initially we collected somewhere around more than 200 samples, but using this panel of 7 microsatellites, we could identify 109 individuals, and these were the diversity estimate for that 109 individuals. The FIS actually represent that inbreeding coefficient index, and which is like we found that our population is considerably inbred. And I was interested to know that how many populations were there in our data set. So we do the Bayesian population structure analysis using structure software, and based on the posterior probability, we found there was 2 populations in the data, and this was the level of admixing and the samples, and the table predicted how that individuals are clustered into different populations. We found that individuals are roughly equally assigned to cluster 1 and cluster 2. This is somewhere around 25%, while approximately 50% were individuals were those which were not assigned to any of these clusters. So now I am looking for the reason why this population structuring takes place.